Genetic engineering involves directly altering an organism's DNA sequence. This process can modify traits or correct genetic defects. Tools like CRISPR-Cas9 are used to precisely target and edit specific genes. DNA comprises a double helix structure containing genes that code for various traits. Genetic engineering enables scientists to modify genes within the DNA structure. Modifying genes can potentially influence physical characteristics, disease susceptibility, and cognitive abilities. Genetic engineering offers the potential to eliminate inherited diseases by correcting faulty genes. Conditions like cystic fibrosis, Huntington's disease, and sickle cell anemia are caused by specific genetic defects. Correcting these genetic defects can lead to healthier lives for future generations. Genetic engineering may extend beyond disease prevention to enhance human traits, including intelligence and athletic ability. Genetic enhancement could potentially increase resistance to environmental factors. Enhancing human traits raises ethical questions about the definition of normal. The potential for creating designer babies is a significant concern in genetic engineering. The central ethical question in genetic engineering is whether altering the human genome is morally acceptable. Concerns about unintended consequences are a key component of the ethical debate around genetic engineering. Genetic engineering may exacerbate social inequalities, creating disparities in access and outcomes. The potential for a slippery slope towards eugenics is a significant ethical consideration in genetic engineering. A potential societal risk involves valuing individuals based on genetically engineered traits. Genetic enhancement could lead to discrimination against those who are not genetically modified. Vulnerable populations may face further marginalization due to genetic enhancements. Widespread genetic engineering could amplify existing social inequalities. Affluent families may access genetic enhancements for their offspring, potentially creating a genetic gap. This genetic divide could further disadvantage those without access to these technologies. The availability of genetic engineering raises ethical concerns about fairness and equal opportunity. Altering the human genome carries the risk of unintended consequences. Modifying one gene could have unforeseen effects on other traits or biological processes. Unintended consequences of gene editing can potentially lead to new health problems. Germline editing alters the DNA of reproductive cells, leading to heritable changes. Somatic editing modifies non-reproductive cells, affecting only the individual being treated. Germline editing is controversial due to the potential for unintended consequences in future generations. Somatic editing offers a targeted therapeutic approach without altering the genetic makeup of subsequent generations. Governments and international organizations are addressing the regulation of genetic engineering. Some countries have enacted complete bans on germline editing. Other countries are developing guidelines to ensure responsible research and application of genetic engineering technologies. CRISPR-Cas9 is a gene editing tool that allows precise targeting and modification of DNA sequences. The guide RNA molecule directs the Cas9 enzyme to a specific gene location on the DNA strand. The Cas9 enzyme cuts the DNA at the targeted location, enabling gene editing. CRISPR-Cas9 technology faces challenges due to potential off-target effects. Off-target effects occur when the CRISPR-Cas9 enzyme cuts DNA at unintended locations. Unintended DNA cuts can lead to mutations and other undesirable consequences. Public opinion on genetic engineering is divided, reflecting both optimism and concern. Optimistic views focus on the potential of genetic engineering to cure diseases and improve human health. Concerns center on the ethical and societal implications of genetic engineering. The debate surrounding genetic engineering involves complex considerations, balancing potential benefits and potential risks. The long-term effects of genetic engineering are largely unknown. Careful consideration of potential consequences is crucial before widespread implementation of genetic engineering. Unforeseen mutations and ecological imbalances represent significant risks associated with genetic engineering. Comprehensive risk assessment and ethical guidelines are necessary to mitigate potential harm from genetic engineering. Equitable access to genetic engineering technologies is crucial for societal well-being. Limited access to genetic engineering technologies can exacerbate existing social inequalities. If genetic engineering technologies are only available to the wealthy, it could widen the gap between different socioeconomic classes. The future of genetic engineering is uncertain, necessitating open and informed discussions. Ethical implications of genetic engineering require careful consideration to prevent misuse. Societal implications of genetic engineering demand responsible development and application. Responsible use of genetic engineering requires continuous evaluation and adaptation.